Thank you for the introduction. My name is Michael Donovan, and today I'll discuss with you some nucleic acid modalities for diagnostic and therapeutic purposes. More specifically, we'll discuss aptamers. So by show of hands, how many of you have heard this term before? Got a couple of folks, good. All right, so I'll do a brief review of the history of them and how they progressed into development right now. So aptamers are a special class of DNA, RNA, single-strand DNA, RNA. They can make targets specific to certain cellular membranes, proteins, and small molecules. All right, here's a typical structure. So you have this stemma loop. It could be a couple of loops depending on what your aptamer strand is. And this is referred to as the binding pockets. An aptamer is it's broken down by aptus, which is a Latin word meaning to fit, plus meros, which is a Greek word which means part. So these are fitted parts, if you will. And they were created in 1990. And they've come a long ways. So the beginning, it was somewhat like the beginning of the development of laser. So we have these neat molecules that we're not quite sure what we want to do with them, but from a scientific perspective, they're quite fascinating. And over time, they've been kind of termed the chemical antibodies. And that's somewhat true. They are able to target certain membranes or other proteins, just like antibodies, but they have a number of advantages that, as the chemistry has progressed for aptamers, have come into fruition and are playing a real role in preclinicals. So let's first give a brief comparison compared to aptamers versus antibodies. So number one is the size. Aptamers being DNA, RNA-based, very small. So the antibodies have these big, bulky structures, obviously because it's a protein. And this can be a problem if you're using it for in vivo purposes, say for tumor penetration, or if you want to really get into some areas that you cannot have such a huge protein crossing, or for cellular uptake, these aptamers play a better role. Also, aptamers can be produced in vitro. And the antibodies you need like an animal model. So mice, sheep, goat. And that raises a number of problems from batch to batch, differences, and also you have to increase the cost quite a bit. So if you want to order an antibody in Kenya from a lab in Florida, your cost of shipment has to be very tight, has to be controlled. It has to get to you within a certain number of days, and then you have to unpackage it to then put it into the freezer or fridge, depending on what your use will be. Aptamers, you just have to give a call to the doctor and whatever lab you're working in or whichever company. He can send you an email with the sequence. You can synthesize it in your lab, purify it, and you have it within the same, with having the same functionality as if it were shipped to you. And that really can decrease the cost and also the time. Having these robots do this and having a line of them, you're able to knock out a lot of aptamers, say within a month. And for public health outbreaks, this is critical. Time is very critical. For a good antibody, you're looking at a few months to get one that then you can validate. Aptamers, you get a whole strain of them. Some of them may be junk, but you can get them in a week and be able to validate right then and there. Also, since they are done in vitro, able to use them in arrays that work magically at high levels. I believe some of you have spoken about the trends of multiplexed diagnostics. So we don't want to only look at 5 proteins anymore. Immunoassays they play a strong role, will continue to play a strong role in certain areas. But let's say if we want to look at 800 different proteins, or 1,000 different proteins, it's a difficult enough challenge trying to get 20 or 30 antibodies working in the same conditions together. Plus you need to have these dyes. Aptamers, they're its own barcode. So you can directly label a protein without having to have a dye via the microarray. And the array community has done a wonderful job at being able to define the DNA, RNA concentrations. Another one, storage. So I briefly talked about this. So with these aptamers, you can keep them at room temperature for a year, for a few years, no problem. Antibodies, that'll be a big problem at room temperature or even at higher temperatures. Even at lower temperatures, you'll have degradation problems. Aptamers, put them on the bench top, 2 years later, you can still use them. Now, if aptamers are so wonderful, why has it taken us this long to then be able to discuss that we're at preclinicals for some interesting applications? And the main aspect was the nucleate degradation. So if you have an unmodified RNA or unmodified DNA aptamer, DNA nuclease will bite that really quickly. It'll eat it up really quickly. So you have a half-life that is just very small. So what the chemists have done is done a number of modifications. On my lab works with, particularly, is the locked nucleic acid. And that's able to increase the half-life up to 53 hours, 72 hours. You can pegylate it, but then you run into some other problems with pegylation. So that's, if you've been up with some of the recent clinical trials, there's a anti-thrombin aptamer that had some toxicity issues, and that was because they added too much pegylation, which is a shame because that's one of the beauties of aptamers is very low immunogenicity. So with the antibodies, if you're on a course of it for a month or a couple months, and you'll develop this immunogenic responses to it, and this varies by people. But since this is a DNA RNA based conjugate, you have very low immunogenicity, so you can really increase these concentrations. And that's kind of the balance that we're playing with. Is one hand, you want the half-life to be high enough that you're able to get to your target, but then also you don't want to add too much modification that then you get some negative immune responses. Now, how do we make these aptamers? Traditional process is CELX, systemic evolution of ligands by exponential enrichment. What does that mean? So it basically means that you're going to start with a random pool of DNA, subject it to positive target, collect what binds to this target, subject it to a negative target, and then throw away what binds to that negative target, and keep on doing this cycle until you have only a few strains that bind to your target. And if you have just a protein, or if you're doing some toxicity reports for out in the field at a point of care to test if there's a toxin in the grain, that may be an okay one. But as you may guess, science and biology is never that simple. So this is a silicon grain, and if you're purifying a protein and using traditional CELX, then some problems may arise that you're binding to the part of the protein that is not outwardly expressed natively on the cell. So what we at the TAN lab developed is a process cell CELX. So this one, you're able to use whole live cells, have the positive cell selection, say CD133 positive glioblastoma tumors, and then here you can have a CD133 negative tumors, just for example. And you do these rounds maybe 10 times, depending on how lucky you are, or depending on the right circumstances. Sometimes if your conditions are really nice, you can just do it 5 times to get some good aptamers. Clone it, sequence it, and then you can perform other applications. Now the beauty of CellSelex is really for biomarker discovery. Biomarker discovery is so difficult because of the discovery aspect. We don't know what the biomarkers are. And CellSelex allows us to target these unknowns. So if we have cancer cells that are differentiated, you can put it at different phases of the cancer cell development and see, all right, which part is how's the protein membrane, how's the proteomics of this cell membrane changing? And then that's when you start to get some fascinating results. Here's just a imitation about the binding of it. Now, please don't think that just because they're DNA, RNA, that this is binding from hybridization. These are 3D binding, so you have these variable regions that form these little pockets against the biomarkers, or whichever target that you have. And these are some aptamers selected for various cancer cells. Now, this is the part of it that's really interesting, is for the past 150 years, we're referring to cancer where it's found in your body. Liver, lung cancer. When you think about it, it makes no sense that that's how we do it. 150 years ago in France, it made sense because we didn't know about proteomics, we didn't know about genomics. But now, we need to improve our lexicon. We need to add the terms HER2, EGFR, PSMA, and others that we will be able to identify from CellSelex and from these biomarker discovery projects. Now this is a really beginning of a large project that we're working on, and it's under the assumption that, all right, you have these cancer cells, and identifying one aptamer for all lung cancer cells or all overly expressed X protein cells can be very difficult because cancer cells are so amorphous. However, you may be able to get some trends going on, and that will give you a better understanding, and then give you a better understanding of which drugs to select for, and which biomarkers make a true biomarker aspect. So this is what we've done. We've taken these aptamers, labeled them with iron oxide nanoparticles, subjected them to each 1,000 cells in a 250 microliter sample volume, and then we submit them under MRI scans, and we checked out the readings. And then over these you can see SGCH is binding to quite a few, but SGCH is a really good CEM aptamer. So this one is really high spikes. Now, when you look at medicine, it's really difficult to get data from your doctor, especially in the US. You go to a doctor and it's such a protected relationship, but also a relationship that the doctor doesn't give out the digital information. And in this age, that's really what we need. We need cross-platform sharing and having thousands of different patients sharing their aptamer or proteomic or whatever type of diagnostics and analysis of the proteomics cancer cells going on, so that we can then be able to gauge, all right, which direction can we go to fully understand this? That's for the past 35 years, we've been asking and trying to understand what cancer is, but made very little growth in the war on cancer. We haven't been asking, how do you control cancer? And this is really what gets me excited about biomarker discovery aspects of Aptamers, is they're able to then be able to look across all these aspects and be able to change the way we look at cancer. This is another slide kind of giving the same idea of having Aptamer library across all other cell lines to see which ones will be nice little targets. Now that's the proteomic diagnostic aspect, but what I'm most interested in is the therapeutic aspect. And for Aptamers and for real cancer therapy, you need to have a multi-pronged delivery and multi-pronged approach to be able to tackle it. You can't just do chemotherapy, obviously, because it's not working well at all. And also, the Aptamers can be used for immunotherapy, siRNA, and also drug delivery. So some of you, as a quick aside, some of you may want to Google John J. Rossi. He does some good work with using Aptamers for antiviral therapy and HIV in specific. He does. He's out in City of Hope in California, and he has some interesting work going on with that. So in addition to cancer therapy, antivirals for HIV, HCV have shown some promise. Let's look at it more towards the cancer aspect. So we have these Aptamers that they are chemical structures. So you can really be able to modify them nicely. You can add, conjugate them to siRNA, conjugate them to different nanoparticles, have drug-loaded nanoparticles, and then that's when you're getting into some neat things. So the siRNA aspect, on one hand, is really difficult to get to the right cells. So we have these spherical nucleic acids that are really cool because you can take a 3D structure for these siRNAs and they just go across all cellular membranes. It's really fascinating that they do this because you think that the differences in charge that will be difficult to uptake. But sometimes you do not want these siRNAs to go to every cell. So what then you can do is have these Aptamer-guided siRNA conjugates. You can also have there's some Aptamer protein conjugates, Aptamer streptavidin, and then also what I'll talk about a little bit later is for photodynamic therapy. But first, let's jump right into one that I worked with this great gentleman called Gui Zhu on building these DNA nanotrains. And this is one aspect that for targeted drug delivery. Now the difficult part is there's really no study showing that chemotherapeutics get a lot of the drug to your target cells. There are different studies showing maybe that doesn't work, the efficacy is very low, but chemotherapy is a very good tool that we can utilize as long as we're getting the therapeutics to the specific cells. So we thought about, all right, how do we bring on just an Aptamer and the chemotherapeutics? And you're looking at that drug volume, that drug load. So if you have one Aptamer to one chemotherapeutic, that probably won't be enough. So these cells are so difficult to kill that having one small doxorubicin to one head structure of your Aptamer will probably not be enough. So what we had was this Aptamer-triggered chimeric. And here, then we added these two M1, M2 strains, which we dubbed the kind of the boxcars, if you will. So this Aptamer trigger is the head of the train. Now these boxcars are are thermodynamically very stable. There's no reaction if you just have them. However, this red string acts as a trigger. So if you look at this, once you put them all together, then you have these boxcars that line up nicely. And we've made some about 400 nanometer, very long structures. You can limit how long this length goes, but they get quite a bit, and you can get some per aptamer, a huge load of doxorubicin. And doxorubicin interpolates with GC-rich sections of your aptamers. So right here, this is very GC-rich, and this, as you see, is the nanotrain. So we subjected these, and the premise, we're still at the beginning stage of this, so we have a mouse model, and it worked pretty well. Now then, so this is some of the results. So we have the SGC8 nanotrains, free docs, and SGC8 nanotrain docs. So SGC8 is CM, target-specific aptamer. Now this is a mouse model that we injected, and as you can see right here, the control, the mouse model, the tumor grows quite a bit, and then for the nanotrain doxorubicin, we do have a significant amount of improvement and reduction in volume size of the tumor. For the C, we have measured the weight also. So this one has to be optimized further, but the weight of it, the cell, free doxorubicin did destroy a significant amount of the tumor volume. However, the mouse also lost a lot of weight, and the survival percentage over days was significantly less. So this is one multi-pronged approach that you're able to use the aptamers for drug-loaded therapy, drug-guided therapy. Another recent project that I worked on is quite interesting. It's using upconversion nanoparticles. So we have what's been termed photodynamic therapy. Photodynamic therapy, in essence, is using light to trigger reaction. So you have some molecules that, if they're triggered with under visible light, will then emit singlet oxygen, and singlet oxygen is, for a cell, is very lethal. So the problem is visible light is a very low penetration. So for biological applications, it's not very helpful to have a light trigger. So we thought, well, what if we take an upconversion nanoparticle that can be excited in the IR region, but also give off visible light, and have this close enough to one of these photosensitizers? What will happen? Will you be able to get it close enough that's able to give off the singlet oxygen? And that's what we did in this setup. So you have at the core of this, it's also a spherical structure. At the core, you have these upconvergent nanoparticles. And to link in it, we have a bio-labeled end of it. This is a G4 structure of just a sequence of DNA that can fold upon itself, causes these quadruplex structures. So this quadruplex structure, we're able to add the photosensitizer, TMPY-P4. And those are able to stay there thermodynamically, tested for many hours, and it did not diffuse in our serum and enzyme-based model. Now, to the head of that, we then added another SGC8 aptamer. And we want this to be able to guide to the cells, and once you're endocytosed, then the mechanism is essentially diffusion. So you got these aptamers, and they're diffused into the cell, near IR region of wavelength, and you're able to excite these upconversion nanoparticles, which then get off the single oxygen. Now this is what we did some focal microscopy to show that it was actually able to get to these target cells, the CM cells, versus the rainbow cells. And as you can see, the CM cells are highly bright, whereas the C and D, whereas the rainbow cells are dark. So it did have some, it was able to differentiate between the CM cells and rainbow cells quite nicely. And after subjecting it to the IR region, the cell depth was quite substantial. These are just to show that the proper binding from the CM cells to the upconversion nanoparticles. So these last two projects they're interesting, right? So the upconversion nanoparticle one, it gets you excited, but it's still a long ways from the applications can be immediately to the clinical trials. However, there are the simple drug-loaded and drug-targeted therapy for aptamers would probably be the first one that will make it into the past clinical trials, but also immunotherapy. So immunotherapy is one aspect of aptamer applications that we're developing in our lab that showed some really good preclinical trial aspects, and that's one combined with the antibodies. So antibody therapy and immunotherapy is all the rage right now, and however it's extremely expensive. So in the US, a treatment of that would be $90,000, $100,000 for one, but that still only has about 15% efficacy, so it's really low. If you use aptamers, then you can be able to use these multifaceted approach and multi-pronged different ways to attack the system. Because that's really what cancer is. We look at cancer as if it's an infectious disease almost, as something you get, but it's something you have. It's a system. You have an inward, a middle part, and an output. And this system is what we have to change. And in order to do that, we must rethink the way that we're treating cancer. Make it more of a not so much a chronic aspect, but something that we give treatment to at the early stages and not just at the end. So the best that we have right now is looking at a patient's cells under the microscope, and it's kind of like a bottle of water. So you take out a few cells, the doctor looks at it and they says, okay, this is a normal cell. And then if it's crushed, this is a cancer cell. And that's the best we have in a lot of clinics right now, which is terrible. We have to change that. So having these proteomics linked up with aptamers will give us a better understanding of what we're dealing with and how we can do it. Also, this will take a different way of how we go to the doctors. How do we get therapy? Right now, we spend most of the cost on the last 2 years of life. After you're at stage 3 or stage 4 cancer, then at that point, it's really difficult to treat. But in effect, we're all cancering. There's this one fellow named Danny Hillis, and he coined that cancer should be not a noun, but a verb. Because all of us throughout our life are cancering at any given time. It's just a broken system, but for the most part, we're able to fix it. So by giving subjective levels of aptamer-based radiation or aptamer-targeted immunotherapy, or aptamer-targeted even chemotherapy, in small dosages at different areas that we're able to kind of boost our immune system and work with our immune system instead of dropping the nuclear bombs that disrupt the whole system, including our immune system that is trying to fight it, then we'll be able to make leads to that and not have it as and be able to then actually make moves to oral cancer. I'd like to thank the members at University of Florida, the TAN Research Group, and then also at Hunan University, the MBL group. So this is where I began my research way back in, well, maybe Paul says way back in 2007, it's not way back. But anyhow, I began over there with Dr. Wei Hong Tan, and he has a laboratory also in Hunan University, Changsha, China. And Changsha, China is central, southeast China. And there's really a remarkable group over there, and they're producing some interesting things. Thank you. Questions?
Yes, please. There will be some reactions to this presentation. I see there, Armand. I prefer that the pronunciation is Hunan.
Hunan, yeah. Hunan is up north.
I say Hanan.
Different place.
Thank you very much. Very interesting talk.
Thank you.
Actually, my question is, because I don't know so much about this. You were comparing to antibodies.
Yes.
In antibodies, you have a specific active site, but here the selection is basically, could be anything, as long as it's specific. Have I understood it right?
Well, it depends on how you set up this selection. So for cancer cells, it's better that we don't really know what it is, because we don't know what's a good active site for. But if you have other proteins or for, let's say, for viruses, then we know the protein envelopes, what it's like, and then that's how we can target it. Or also, I worked with glioblastoma tumors that we knew that they were heavily CD133 expressed. So for that, there'll be the CD133 that's targeting, and you can arrange the selection for that way. But the actual site that it binds to, we can't really control that too much. We can control that it goes there. But it's a structure that it kind of, it binds to it, but also it can have a little bit of overlap to a different part of the cellular membrane that it's binding. So that's the kind of the black magic of it.
I'll make your life a bit more simpler, because here what I was thinking was point of care diagnostics.
Yes.
Shelf lifetime was over years, which means people doing protein-based, say, ELISA or anything about that. Can you comment on, if you want to do it for a point of care, then you would have to have it on a solid support. Is it possible to design them so that they're active? So would you know which side of it is active and then can one design?
Oh yes, so they have, you're able to do the sandwich-based ones, and it actually for sandwich-based ones, with the antibodies still are a little bit better. Do you have more advanced systems that kind of that go on maybe the piezoelectrics that will be able to give the sensitivity much lower. And that's still, you could do at a point of care system, but it'll be a little bit more expensive, but on the long term, it could work out because it can be all at room temperature, and you can then deploy it to very rural aspects.
Thank you.
Thank you.
Okay, one more.
Thank you, Michael, for your presentation. Just to continue with the previous inquiry, I think it will be interesting to have a percentage of specificity of your aptamer application. Moreover, maybe have the coefficient of affinity. My question is to know, is there any possibility that instead of binding to the target cells, it binds to another cell, maybe leading to a problem? Thank you.
Oh yes. So this is what you have to do throughout the validation aspect. And let's go back a few slides. So right here, these cells, these are different aptamers, and they, in fact, are binding to to some other cells. So let's look at HC807. So this is a very good small cell carcinoma aptamer. However, it also binds to adenocarcinoma and then IMEA liver cancer. And there could also be some binding to normal cells, and then that's why we have to have a huge library to make sure that it's not binding to it. And that's what you do during the validation process. So when you're having it with your cell select part, you do get maybe panels of 20 aptamers or 10 aptamers that could be worthwhile. But then you look at it, okay, are these binding to other cells? And if they are, how much, to what degree? But also when you're looking at it, it's kind of a game, it's a balance. So with the regular chemotherapy, it just goes everywhere. So if it's going to maybe partially to some cells, but mostly to these cells, then you're still much better in your much better situation of winning that war aspect. But it's one that you have to do from the beginning, or not from the beginning, but at an early stage. And what a lot of doctors and researchers are recommending now is to not wait till we can actually see the cancer cells. So we know a lot of biomarkers. So if you have a low dosage, then you're able to then maybe pop it off a little bit. Pop it off is the professional term for that. So give it just enough chance that your immune system is then able to take over the job. So it's not a magic bullet by any chance, but it's enough that you're able to have enough applications of it to be able to make some good headway.
Okay, Professor Massi.
Only one short question. Very nice talk, by the way. So the affinity of the aptamers cannot compete with the good antibodies, or they are in the range of picomolar or just lower?
So it depends on that. Most of them that we work with are low nanomolar. Some are in picomolar range, but most low nanomolar. So if you do maybe an immunoassay, just 1 versus 1, then the aptamers are quite lousy in that area. So it really depends. I don't think the aptamers will ever truly overtake antibodies, because there are a lot of aspects that antibodies are very good at. But there's other aspects that aptamers have been able to mitigate some problems that antibodies have. And then we have a really large scale of over 100 plus targets, 300 plus targets, where you want to look at more of a metadata analysis, and the aptamers will be very good for that.
So Larry Gold, the company, I don't remember the name of the company, they have a array with around 1,200.
Yeah, so Larry Gold, he's running SomaLogic. And they have these things called Aptamers. And those are very interesting.
But in the late 20 years, people have tried to get into the therapeutic, but as I am aware, it's only one that is in clinical trial.
Yeah, so Larry Gold, bless his heart, but so the real patents for aptamers have gone off only a few years ago. So now most of the patents have expired on aptamers, and Larry Gold's companies had most of them. And he put most of his emphasis on the Summerberg aspect, and that is actually coming to fruition, and he's doing some really good collaborations with some hospitals and making some good findings for biomarker and then also for more proteomics. But that also then limits other players from entering. And the only FDA-approved drug right now was for Macugen, which is literally a stick in the eye. So it's not a very good, that one for antibodies, actually, the antibody drug did work better. But for cancer therapy and so prostate cancer, they have some really interesting findings that right now we'll be able to go to the clinical trials. And one other aspect that I like to add is a problem in the aptamer community called the thrombin effect. So thrombin is one of the most popular aptamers in the beginning, and you can buy it online for $30. So if you're an analytical chemist and you want to do a new research project, and you can just buy this one for $30, tweak it a little bit, and then you get a paper, instead of actually then looking at a different aptamer, going through approval process, raising funding, then going through FDA clinical trials. You know, a lot of the researchers then just opted to get the thrombin one. So it's a so easy aspect of creating these aptamers that it in of itself actually kind of inhibited the growth of it. All right, thank you guys. Oh, I think we're over time. One quick one. Okay.
Okay, thank you for this interesting presentation. My question has to do with small molecules.
Yes.
The problem with antibodies is they're really terrible at picking up small molecules like some carbohydrates, steroids, nucleotides,
Yes.
lipids. I'm hopeful, sort of. I mean, you can't get an ELISA for a lipid biomarker, for instance.
Yes, I'm glad you brought that up, because smaller molecules are something that Aptamer is able to target for. So if the antibody is very difficult if there's no immunogenic response to it, right? And then, but also the Aptamers have been shown that even a difference of one methyl group be able to differentiate for these clean targets. So there are a number of papers on small molecules. I personally haven't done much research on that, but I'll send you over a few doctors or reports that I know are credible.
Thank you. I appreciate